Hi, my name is Civetti, and my group tested the effect of pH on trypsin function. This experiment studied trypsin, which is an enzyme found in the intestines. An enzyme is a type of protein that acts as a catalyst, meaning that it brings about a certain chemical reaction. Enzymes speed up the rate of the reaction by lowering the activation energy for said reaction to occur. In living organisms, enzymes are needed to speed up reactions in the body as well as regulating the reaction rates. Trypsin is a proteolytic enzyme, meaning that it breaks down proteins. To do so, trypsin speeds up the hydrolysis of peptide bonds on the carbonyl side of amino acids, lysine or arginine, basically cleaving at the bond. This process is needed in order to digest dietary proteins in a timely manner. In animals, trypsin is formed in the pancreas and then is converted to active trypsin in the small intestine, except in bacteria, trypsin is secreted in order to break down proteins. So our question for this experiment was, how does the pH affect the function of trypsin? We hypothesized that at a constant temperature of 50 degrees Celsius, trypsin will reach peak function at a pH of 9. This was based on groups who studied trypsin function under different temperature conditions. About 50 degrees was the optimal temperature for trypsin activity. The materials needed for this experiment was reaction buffers of pH 7, 8, 9, 10, and 11 with a salinity of 0.15 molar NaCl, trypsin, azocasin, 10% TCA, an incubator, a centrifuge, a spectrophotometer, and a 50 degrees Celsius heat bath. Add 450 microliters of pH 7 reaction buffer to 3 microcentrifuge tubes. Then add 500 microliters of azocasin followed by 50 microliters of trypsin. Do this 3 times for each pH. There should be a total of 15 tubes. Then incubate for 20 minutes at 50 degrees Celsius. After incubation, add 500 microliters of 10% TCA to each tube. Centrifuge the tubes 3 times, twice at 12,000 RPM for 2 minutes, then once at 14,000 RPM for 2 minutes. Transfer 700 microliters of supernatant from each tube to a separate cuvette. Blank the spectrophotometer at 440 nanometers and read the absorbance for each cuvette. Then, to prepare the negative control, follow the same protocol except do not add any trypsin to the solution. There should be a total of 30 samples for this experiment. TCA was added to stop trypsin from reacting and also precipitates undigested substrate. By centrifuging the reactions, it forms a pellet which removes undigested substrate. The supernatant contained a digested substrate, which is used to read absorbance. Azocasin was used as a substrate, which is derived from casein and sulfanilamide groups that are covalently linked to peptide bonds. The orange color from sulfanilamide is needed in order to read absorbance at 440 nanometers. Our control without trypsin was chosen because it would not have any type of reaction, thus giving us a baseline for what to expect. As described previously, negative controls were prepared according to the same procedure, just without trypsin. This measures the absorbance of azocasin when not degraded by the enzyme. 3 replicates of the negative control were used to provide multiple sources, render good overall average, and to have multiple options in case one attempt failed. In this bar graph, we have displayed our averages, standard deviations, t-tests, and the x-axis displays pH. The y-axis is described as absorbance. As you can see, there is a valley at pH 9 and 10, though we hypothesized that there would be a peak. A t-test is used to show the significant difference between 2 groups. Several t-tests were performed to compare average absorbance at different conditions to the average absorption at pH 9 and to determine if values differed significantly or if variance was due to chance. All conditions except pH 11 showed a p-value of less than 0.05, therefore results were significantly different. At each pH, there was a standard deviation of less than 0.0364, showing very little variation between absorption values. This experiment was executed with high attention to detail and precision. We feel it was adequate enough to test the hypothesis. The results, however, did not support our hypothesis. Considering the results from our first experiment, which show peak enzyme activity at a pH range from 7 to 9, as well as class data which showed optimal trypsin performance around temperatures of 50 degrees Celsius, we had hypothesized that at a constant temperature of 50 degrees Celsius, trypsin would reach peak function at a pH of 9. However, our results showed that trypsin had the lowest function at a pH of 9 and 10, which did not support our hypothesis. If we could conduct this experiment again, we would do so with the same attention to detail. However, it would be conducted to provide further backing to either our first or second experiment results. Because our conclusion found that trypsin functions best at higher pH levels at the temperature of the small intestine, to grow bacteria, lower pH levels would be used because bacteria grows best in acidic environments.